So let's go over chapter 8. Okay, so we began just by giving an outline of the vessels that we're dealing with. And the important thing to remember is that these vessels are not the same throughout the circulatory system, all right? So here, leaving the heart, we have the arteries. And the definition of arteries are those vessels that bring or take blood away from the heart, okay? So more specifically, they are under higher pressure. They, in general, have thick walls, and there are no valves, all right? So these arteries become progressively smaller as we get further and further from the heart, and eventually they're called, instead of arteries, they're called arterioles. And I also said that we have a few different types of these larger arteries, and I'll get back to that in a minute, but we have these strong muscular arteries, and we have elastic arteries, and I'll go over that in a little bit. Now, eventually, our arterial system will become small enough, and we have these vessels called capillaries. Now, the capillaries, they are single cell thick, all right? They have no extra layers, and that's important because this is where we have the exchange of nutrients, gases, and wastes between the tissues and the blood. And, of course, these are under very low pressure. Now, on the other end, carrying blood back towards the heart, we have the venous system, so veins. So veins, what do? They carry blood back to the heart. And again, they're going to start off small, and they're going to get progressively larger. So the smallest of these are called venules. And these vessels are under much lower pressure. They are thin-walled, and they have valves. And very importantly, they're able to store blood. All right. Even though the blood is still flowing, the venous system is our storage location. All right. And I'll go back over that. Now, in terms of the layers of the blood vessels, the layers are called tunics. And we can compare and contrast the veins and the arteries here. So if we start in the middle, the tunica interna, or the innermost layer, is, of course, your simple squamous epithelium. That is given that special name when we talk about the circulation. That's called the endothelium. And next to that is going to be the tunica media. And so after we have the epithelium on the basement membrane, we have the tunica media. And this is where we have our muscle. So our M and muscle, so middle layer, media, muscle. Okay, so here we have are very often two layers of smooth muscle, circular and longitudinal. And then on the arterial side, we can also see that we also have this possibility for these elastic layers. So there's some elastic fibers that can also be interspersed in here if we're talking about elastic arteries, all right? We won't have that on the venous side. And that's very important, which I'll go over again. However, and I neglected to remind you that on the internal layer, on the epithelial layer, on the venous side, we have valves, okay? So we have valves, and the valves don't exist on the arterial side, all right? Now, the outermost, or the most external layer, the tunica externa, is basically connective tissue, all right? So we have that on the outside, helping to hold these vessels in place. Now, if you compare arteries and veins, and we can compare similarly distant arteries and veins very often in the tissue, they'll be close by each other. You can see here that the arteries are clearly have smaller diameter. They are thicker. The veins, the diameter is much larger. They tend to be a lot more floppy in appearance. And you can see that they have a thinner wall, all right? Now, in between both, of course, we have the capillaries, and again, that is your simple squamous epithelium. The drawing is kind of misleading. Very often it's just a single cell all the way around here, on the very smallest ones, and then they're sitting on a basement membrane, because any epithelium needs a basement membrane, and that's what it's sitting on. Now, as you can tell, as you get further and further from the heart, the vessels get smaller and smaller, and the layers get thinner and thinner, including the muscle layer. And eventually what you're going to have on the arterial side, the very last muscle cell. And that very last muscle cell actually has a function other than just being a muscle cell, and that is that it acts as a sphincter. So it's called the precapillary sphincter, and it can be regulated to control how much blood is going to go into the capillary bed, all right? So the brain can control that. And we can actually have entire capillary beds shut down for a time, all right? And if that happens, the blood will flow directly from the arterial to the venous side through something as a vessel called the metarterial, all right? So that's what's being shown down here. Now, the brain can, or the body, I should say, the body can control how open and closed these vessels are. So vasoconstriction, or the closing of the vasculature, and the vasodilation, the opening of the vasculature, is controlled in 3 different ways. First of all, neuronal control, all right, so that would be top down in the brain, right? We also have hormonal control of this process. And there's also local controls, all right? So locally down at the capillary bed level as well, all right? So there's factors produced by the cells in the tissue that will help regulate things more directly in those tissues, all right? So what do we mean by constriction and dilation, and why is that important? Well, if we have a constriction of the vessels, all right, what's happening then is that the diameter of the tube is getting smaller, so we're constricting it. When that happens, you're clearly having less blood flow through that area, but you're increasing the pressure because the amount of blood at any one time is basically the same. Okay, there are ways to change blood volume, but a little bit past the scope of this course. But in general, you have your certain amount of blood, 5 to 6 liters, depending on your size, and that's your volume. So if you constrict the vessels, you're going to increase the pressure. Now, conversely, if you dilate those vessels or let the diameter get larger, okay, you're going to have increased flow and you're going to have decreased pressure. All right, so that's exactly the opposite. And this is basically how these things are controlled. Now, the next thing is to think about this pressure gradient. And I don't think it's surprising to you that the highest pressure is where we have the blood leaving the heart, so at the aorta. And as we go further and further from the heart, the pressure gets less and less and less. Okay, so we'd start off with, here it says, you don't need to remember the numbers, but you can start off with something like 93 millimeters of mercury at the heart, and it can go down to just a few, 2 or 3 by the time the blood returns to the heart, the vena cava. All right. So the blood is flowing down a pressure gradient. In everything in biology, if it's working passively, it always seems to be from high to low. So high to low pressure, all right? And the blood is no different. Now, this is where we start talking about the elasticity of these arteries. Now, the elastic arteries versus the muscular arteries. The muscular arteries are largely controlled, the diameter is controlled by the brain by controlling the smooth muscle constriction and dilation. However, the elastic arteries, even though they have muscle, the brain doesn't focus as much attention on those because the diameter of those vessels is really mostly reliant upon the elasticity of the vessels. So in these vessels, like I had mentioned before, we have these elastic fibers. And I mentioned in class what would happen if I take an elastic band and I stretch it and then I let go, right? And everyone knows that when I let go of the ends of my elastic, that it will bounce back. Well, the exact same thing is happening in these thickest arteries. These expand and contract and move with the contraction and the relaxation of the heart. So they actually carry the energy from the heart and push the blood forward in that way, okay? So vasoconstriction, vasodilation doesn't tend to happen in these largest arteries. It occurs much more so in the muscular arteries, the arterioles, venules, and the veins, all right? Now, So let's think about these large elastic arteries. So if we're looking at our left ventricle, right, of the heart, it contracts. So it's pumping blood out of the heart. And when it does so, there's the semi-lunar valve. It opens up and the blood is pushed out into the aorta. All right. And of course, this is an elastic artery. So what is it going to do? It's going to expand. It's going to stretch, okay, with that pressure. Now, the ventricle is going to relax. So when that ventricle relaxes, there's going to be some back pressure, right? And what ends up happening is that's going to snap our valves shut. And now where is that blood going to go? Well, it can't go back into the heart because the valve is closed. So that energy now is going to be pushing from the elastic recoil, is going to push that blood forward into the body. Okay, so that pushes that blood out to the body for just simply by that elastic recoil. All right, we don't even need to worry about vasoconstriction and vasodilation. All right, so this is what I mean by this. All right, now you can imagine that if there is a defect in that elasticity, however, or with aging and as things become a little more less elastic, these elastic arteries can be subject to something called aneurysms. So these are these defects in these arterial walls, and they start to balloon, or sometimes they'll bulge inward, okay? And that will cause even other problems. And this happens slowly over time. It's not an instant thing, at least not that I'm aware of, okay? And then with time, these vessels can rupture. And one of the main places this happens is in the aorta, because that's where we have the highest pressure. And if the aorta ruptures, then it can lead to very quick death, right? Because all this blood is just going to be pumped out. There's no way your clotting mechanism is going to be able to keep up with this. So here you can see why these are so fatal. Sometimes there are symptoms that are warning signs. I had mentioned in class that my father had this, and fortunately, we were able to know because he would faint, because the pressure would drop so dramatically very often. If so we were able to tell that there was something not right, and they checked him. And they realized that there was an aneurysm, so they actually went in and repaired that. He also had a valve issue, so they did both surgeries at the same time. So they repaired the aneurysm as well as the valve at the same time. So that's what can happen. Now, the capillaries, all right, so moving forward with that, here is where we have the exchange of gases, all right? And what happens here is that since we have thin walls, the gases and nutrients and wastes can be exchanged very easily. Okay, we don't have those extra layers, so things can happen pretty quickly, right? This is largely by diffusion. And we also have to lower the flow. Okay, so when we talk about this, and this is where I brought up this idea of the delta, right? We know that if we have a delta at the end of a river, that the flow of the water going through down that river is going to slow immensely once it hits the delta. And the reason that is because we've increased the surface area that is exposed to the water. And the same thing happens with the capillaries. So the velocity of flow is going to drop dramatically, as you can see here, over these small vessels. And that's because as we go through, we've got the same amount of volume, but we've increased the surface area. And increasing the surface area and slowing the velocity gives more opportunity for exchange. All right, so that's exactly what we would like. Now, we have different types of capillaries. All right, the most of the capillaries are the continuous capillaries. And these have a smooth basement membrane, fully intact basement membrane, and all of the cells are holding on to each other quite well. All right, so that's why they're called continuous. So we have continuous basement membrane, and we have continuous epithelium. Now, the next kind is specialized, the fenestrated capillary. And the reason it is fenestrated is because the cells actually have sort of like little pores that go through them. They're called fenestrations, these little pores. So they kind of look like they've got holes in them, all right? And, but the basement membrane is still continuous, all right? Now, where do we find these fenestrated capillaries? Well, we find them in places like the kidney, all right? Because they look kind of like a dialysis membrane, all right? So that's where we find those. The last type is called the sinusoid, and these ones, you can see that there's holes in the cells, between the cells, holes in the basement membrane. So you can see that blood can get out quite easily. Now, the organs where we have these kinds of capillaries would be the liver and the spleen. So these are very red organs. And these are places where we actually want blood to come out, all right? This is where we recycle all of our old red cells, get rid of debris that we don't need. And also, especially with the liver, the percolation, we see the blood actually percolates through this tissue, and the tissue is structured so that it doesn't let blood go all over your body, okay? Because we've got sinusoids there. The blood is percolating through here, and this is a really perfect way in which to get nutrients out of the blood very quickly and wastes and put wastes in. So very often we say that the liver is sort of the body's factory, right? Because this is where we're making things and changing them around, and we're also breaking down old stuff, right? And this is one of the reasons that the sinusoids would be perfect for this organ, all right? Now, the next thing to think about with these capillaries is the change in pressure on both sides of the capillary bed. And this is something that sort of drives a lot of processes in the body, and also a lot of disease processes when it doesn't work very well, all right? Now, on the arterial side, we have higher pressure, and on the venous side, we have lower pressure, okay? Now, what ends up happening is this drive to keep water in the blood. So remember that the percentage of blood that is watery is like 90%, right? So 90 to 92%, depending on who you're reading. Now, clearly, water wants to move from high to low concentration. So osmosis is going to be wanting to be working here, and it's going to want to, the water is going to want to leave the vessels, okay? And indeed, that's what happens, right? So the process of this water leaving the vessels is called filtration because it's going through a membrane, right? So remember, osmosis is the movement of water or solvent from high to low concentration, right? And in our body, the water is the solvent. And that is indeed what's happening. So on the arterial side, we're saying that the water is filtering because it's going through the membrane. And the thing that's driving that is the tissue salts, right? You've got a lot of ions in the tissues, such as calcium and sodium and chloride and all of these things, and that's going to be pulling the water out, right? And it's also lower concentration, right? That's why it's lower concentration. And the other thing that's going to be pushing water out is the pressure from the heart, right? So literally that pressure is going to be pushing the water out. So these are the two things that are happening mostly on the arterial side. Now, to fight that, and clearly we must be fighting it efficiently enough for the blood to stay watery, is absorption. And this would be due to the back pressure of the tissues. So the tissues are exerting a pressure back on the blood, right? And that is one thing that's going to push the water back in. But then we remember we have our albumins, and those are those large proteins in the blood that are going to pull water back in or keep water in the bloodstream, okay? So we have a conflict or a balance between filtration and absorption. So one is actually winning, and that would be the filtration side. So in the long run, more water is going to be coming out than is going to be being drawn back in, okay? And with this water comes our nutrients, our oxygen, and all of this, and our wastes and all of that go back in, all right? But we clearly can't have this situation. It works out to be about 3 liters a day that filters out, and that's quite a bit. So somehow we need to put that fluid back in. Okay, so remember what's filtering here is plasma, right? That's what our fluid is in our blood. So it's plasma that's filtering out. So now we have to somehow get that fluid back in. Now we don't have a pump out here, so we can't do that. So how do we collect that excess fluid? Well, this is where we have another set of vessels, which are called the lymphatics. So the lymphatics are filled with fluid. It's called lymph, but its origin is plasma, right? So all the fluid that we have in our tissues and the fluid in the lymph is actually originally plasma. And these lymphatics are very similar to veins, all right? But they're not connected to the heart. So there's no circulation here. This is just a set of blunt-ended or blind-ended vessels that are matched up well between arteries and veins, and they collect this excess fluid. They don't circulate red cells. It's just this fluid that's coming in here, all right? Now, how does this work? Well, these capillaries that we have on the lymphatic end, they're blind-ended, so they're not connected to the heart. They have sort of a special overlapping structure, and they're anchored in place so that if they swell out, they're held together. But the overlapping structure allows for water or fluid or plasma, however you want to think of it, is moving to go in but not back out. So they sort of like a valve almost, and it almost prevents fluid from leaking back out if the pressure goes up inside this vessel, all right? So this flow is one way. It's going to go back towards the heart. Not going to the heart. I just said it's not connected to the heart, but it's going to go back towards there. So this fluid has to come back to the blood in some way, okay? So this lymphatic fluid, it's kept directional. It's got valves to prevent it from going backwards, okay? So the valves help it flow directionally. And as it makes its way back towards the heart, the lymph fluid is actually going to pass through the lymph nodes, okay? So the lymphatics are critical to your immune system, because what's happening here is that this fluid that's being filtered and it goes through your tissues, it can pick up whatever's in the tissue. And if there's some sort of an infection of some kind or something that doesn't belong there, as this fluid filters through your lymph nodes where your B and T cells are waiting for a call to action, it will then educate those lymphocytes and help them mature so that when they can then leave the lymph node and go out into the tissues and do their thing, okay? And then go out via the bloodstream. And then some of them can travel through the lymph as well. We have no red cells here, all right? Okay, nothing like that. So here we have this perfect system where we're collecting information through this fluid from all over the body, and it's coming through these lymph nodes to help us fight our infections. So your lymphatics not only are important for returning fluid, filtered fluid from the blood back to the circulation, but also to help you with your immune surveillance, okay? Then we also, of course, have lymphatics in the digestive system. We have them all over the place. We've got lots of capillaries, as you might imagine, in your digestive system, because that's where you're collecting all of your nutrition. And here, the lymphatics also have a very important role, and that is to collect fat and bring it into the bloodstream. The way fats are processed into these structures called chylomicrons, I'll talk about it later when we do digestive, but these fats are too large to fit into the bloodstream, but they are able to fit through the endothelial cells of the lymphatics. So in the digestive system, these lymphatics are actually called lacteals, and they collect these fats. The fats are in these structures called chylomicrons. We'll go over that when we do digestive, so you don't need to know that term yet. And the lymph here is actually called chyle. It has a special name. It doesn't look like the plasma appearance of the other lymph in other places of the body because it has this fat. So this lymph fluid plus fat is called chyle, and that's particular to the digestive system, all right? Now, where does all of this lymph go? I told you that it has to drain somehow back into the blood or back into the circulation. And it actually deposits itself into the subclavian veins, the right and left subclavian veins above the heart. So here's where these veins are, right here, and here's your heart. And there's a duct here on each side that will allow for the lymph to flow back into the circulation. And this is how we keep this system running. And this is also, where we'll recycle some of this fat as well. Now, oh yes, the other thing here is that, curiously enough, the left lymphatic duct, okay, you can see it here, it's called thoracic duct, actually drains most of the body. And I'm not sure why this is, I don't know if anyone knows why, but for some reason, the left collects almost all of the lymph, whereas the right duct, you can see here, it's not shaded in green, it's just basically the right arm, the right side of the head, and a little bit of the right side of the trunk, okay? But all the rest is going in on the left, all right? Now, how is this lymph circulated? Well, the way it's circulated is with these valves, but the rest of the flow is very similar to how we circulate the venous system, right? So here we have very little pressure now in the venous system, but we still have to get the blood back to the heart. So also in the venous system, just like the lymphatics, we have valves, right, to keep this one-way flow going. Now, the way we push blood through here is also through skeletal muscle contraction, right? So that is going to help. And there's also that negative pressure in the thoracic cavity, right? So the thoracic cavity is actually vacuum, right? You know, if you puncture the thoracic cavity, a lung can collapse because we've broken that vacuum. So that negative pressure will help pull the blood and the lymph back towards that area. Okay, so how does this work? Well, here you can see we've got our valves, right? And then we have the muscles. So you imagine the muscles contracting. So you're doing this kind of an exercise here. And as they contract, the valve is going to make sure that the blood continues back upwards towards the heart. All right, so this, as you can see, a valve here. Now you can already look at this, and if there's a lot of pressure due to gravity, and that's going to bring me to my next point, things can happen such as varicose veins, right? So here we can have this situation where we have these varicose veins, and that's when these valves become compromised. They don't close properly, and the blood tends to pool in the extremities, very often around the ankles. And varicose veins can develop into something very serious. The tissue becomes necrotic. You can lose your limb because of this. If this gets bad, you can see how necrotic tissues here, or damaged the tissues are here. And it can lead to death because it will become infected and septic. And this also comes back to this idea of the flow of the lymph, right? I mentioned this in class, that if you have a patient that's bedridden, massage is a very good thing, because not only does it help the circulation of the blood, but it will also help the circulation of the lymph. And remember, the lymph has that double role, in that it's also helping to educate your immune system, especially if this is a person who's in the hospital. That will help them fight their infections if we can let the immune system know that there's something going on here. So it's very important to keep that flow going. All right, so massage is a very good thing, especially for bedridden patients, and just to keep things circulating, okay, amongst other benefits. All right, I usually have some massage students in my class too. Okay, now remember I had mentioned that one of the jobs of the venous system is also to store blood. And how does it store that? Well, because there's some different properties about the veins. So first of all, we have more veins than we have arteries, all right? Now, for example, in your arms and your legs, for every 2 veins, depending on where we're at here, but in some cases, for every 2 veins, you have 1 artery, all right? So you have a deep vein and a superficial vein. And then the artery is just sort of in between that. The other thing is that, as you saw, the veins tend to be bigger, right? So they can hold more blood. And the other thing is since they are not elastic, they can distend, all right? So they can expand, all right? So if there's extra blood, they can sort of expand out, all right? And then when needed, they can be constricted by vasoconstriction, and then we can deliver more blood back to the heart, okay? Now, keeping in mind this is a closed system, so if now you're going to increase the flow back to the heart, you're going to have to make the pump work faster in order to compensate for that. So this is a way of also increasing our heart rate, okay? So there's a lot going on here. Now let's get into the heart. So what's happening here, and I drew this on the board, but we have, and I always start from the venous side, okay? But it's a circuit. It doesn't really matter where you're starting, all right? Now, what you need to know about this is, for this course, is the flow of blood. You need to know that. If I tell you that the blood is now in the right ventricle, you need to tell me where it came from to get there and where it's going to go to from there, all right? So you kind of need to know this, all right? So what happens here is that the deoxygenated blood from the body is going to travel through the vena cava into the right atrium, okay? Now, between the right atrium and the right ventricle, we have a valve, which is a tricuspid, has a, oh my God, here I am. This is the right AV valve, atrioventricular valve, okay? And this one, then from here, okay, we're going to allow for, and it's like, it is a tricuspid, the tricuspid valve. I have a distraction here at home. So from the right atrium through the tricuspid valve into the right ventricle. And then what's going to happen is that when the right ventricle contracts, it's going to pass through the pulmonary valve, and the blood is going to flow through the pulmonary arteries. So first we have the pulmonary trunk, but we go to the pulmonary arteries to the lungs, where we will pick up oxygen, all right? Then from the lungs, it's going to come back, all right, through the pulmonary veins, right? So pulmonary arteries, even though it's deoxygenated blood going to the lungs, all right, because arteries leaving the heart, veins coming back. So here's your the one set of veins where we have oxygenated blood coming back to the heart. So pulmonary veins coming back to the left atrium. Then from the left atrium, blood is going to go through the bicuspid valve, or the mitral valve, that's the common name, mitral valve, into the left ventricle. So left atrium, bicuspid valve, left ventricle, and then it's going to be pumped through the aorta, through the aortic valve, okay? And then it's going to go out to the body. Now the pulmonary valve and the aortic valve, these are your semi-lunar valves, all right? So these then out through the aorta to the rest of the body, okay? So elastic arteries, muscular arteries, arterioles, capillary bed, venules, veins, and then right back into the right atrium, okay? So I sort of color-coded this. So blue would be deoxygenated and red would be oxygenated blood. All right. All right. So remember your valves as well, right? So you have your semi-lunar valves going out from the heart, your tri and bicuspid valves between your right and right atrium, right ventricle, or the left atrium and left ventricle, respectively. Okay. So remember and go over that. Draw it out. Okay. If you draw it out, it's not such a big deal. It doesn't take very long. You saw it took me less than 5 minutes in class. All right. Now, so if we look at this, look at these valves a bit more carefully. So we have the AV valves, so atrioventricular valves, right, between the atria and the ventricles. On the right side, we have the tricuspid. And on the left side, we have the bicuspid, or the mitral valve. And I'm stressing this because if you go to the doctors, they're still going to call it the mitral valve, right? And I know that because my father was in the hospital, and that was his valve that he had issues. One of the valves he had issues with. He had different issues with different valves, but the mitral valve, it's still called that. All right. And the semi-lunar valves are the ones pulmonary. That's the one going out to the lungs, with the pulmonary trunk, pulmonary arteries. And then we have the aortic going out to the aorta, all right? And remember, these valves have different structures. They work a little differently, okay? Now, important to remember that we have these two sets of valves. We have the AV valves and we have the semi-lunar valves, all right? So the AV, the atrioventricular valves, are between the atria and the ventricles, okay? Tricuspid on the right, bicuspid on the left, okay? And these two work simultaneously. So they open and close at the same time, all right? They open and close at the same time. Then the semi-lunar valves, okay? So these are going to be between the ventricles and the arteries. We've got the aortic and the pulmonary valve. And these likewise will open and close at the same time. So when you have your heart sounds, the first heart sound is the AV valve. Those two valves closing, snapping shut, that's your first sound. And your second sound is when the other two valves close, all right? And they open and close opposite. So when the AV valves are open, the semi-lunar valves are closed. When the semi-lunar valves are closed, the AV valves are open, okay? So you could sort of think of the heart as right and left pumps, or you could think of them as top and bottom, all right? So there's two ways of thinking of the heart, all right? So these cuspid valves, they have a really interesting structure, all right? So they open up and they're relaxed, and you can see that the cusps of these valves are held by these little cords that are called chordae tendineae, or heartstrings, all right? And when the valve shuts, right, and this is going to be high pressure, think of those ventricles pushing now blood to the body. There's going to be a lot of pressure. So when these chordae tendineae are very important because they will keep the valve shut under that high pressure, right? And that will then allow blood to not go back into the atria, but out to the body, all right? And these chordae tendineae are held by muscles that contract as well when those ventricles contract, and sort of pull that down, pull those cords down and help keep that valve closed. So there's very high pressure here. And this is actually the way it looks, okay? If you get to A&P and you do a heart dissection, you'll see this really nicely. You'll see these cords, the chordae tendineae and the papillary muscle. All right, and here is your AV valve. And this one apparently is the tricuspid. You can't tell from this dissection which one it is, at least I can't. Now, the semi-lunar valves are different. They don't have chordae tendineae, all right? So these guys, they have these sort of pouches. And what happens here is that when the ventricle relaxes, right, and there's that sucking back pressure, right, wanting to pull that blood back into the heart, these things then snap close, and these things are like sails, right? So the filling up of these things with blood, as the blood tends to want to flow back into the ventricles, as these fill up, that keeps these guys closed. So we don't need chordae tendineae and we don't need papillary muscles, okay? And there's about 3 cusps per valve, so these close up, all right? And then when these close up, they fill up with blood. The elastic artery, when it bounces back, is then going to push the blood out to the body, all right? Now, you'll play with a little bit with this in the lab, but it is possible to place the stethoscope in different locations on the chest to hear the different valves. I showed this, if you're in my lab, I have the same slide, but it's kind of interesting because if you look at the location of these different valves, so here green, red, purple, blue. So this valve is here, but the best place to hear it is down here at the apex of the heart. It's kind of freaky, right? So that was the other thing that I didn't mention here, the apex and the base of the heart. It's kind of weird because the apex is here. This is considered the tip of the heart. So it's the apex. I know it doesn't sound quite right. And the top part is called the base, all right? Okay. Now, of course, you have heart muscle. It's very thick. All right, we're going to go over the layers of the heart in a sec. So the heart has its own circulation, and that's called the coronary circulation. And this is the part that gets obstructed during a heart attack. All right. So these vessels are very important to feed and bring oxygen to the heart muscle itself. And this is really important, as well as taking wastes out. Right? Because even though the blood is flowing through there, this is very thick tissue. So we need capillaries and we need a blood supply just for the organ itself. So we have the coronary circulation. We just mentioned we've got cardiac vein and the left coronary artery. So there's names for these vessels as well. For this course, we don't really need to worry about, you know, what the names of all of these vessels, but it's important for you to realize that these vessels all have their own names and it's its own circulation for the heart and how it works. And it does work with the contraction and the relaxation of the heart as well. This wouldn't be surprising, all right? Now, remember the heart is placed in the thoracic cavity, right? And it's within its own little body cavity, right? And remember there's a serous membrane around that, right? So that is our pericardial membrane, all right? And remember it has two layers, all right? So if we go from the outer part and we work our way into the heart, all right? So the very outer part, we have this thing called the fibrous pericardium, which is a very thick layer of connective tissue that places the heart in exactly the right position. And the parietal layer of the serous pericardium is continuous with that. And then, of course, we have a fluid between the two layers of the pericardium. So remember, the serous membrane secretes that liquidy fluid or watery fluid. And on the other side, we have the visceral layer of the serous pericardium, all right? Now, that is considered part of the heart wall, the visceral layer. And then we have the middle layer, which is called the myocardium. Okay, so remember, every time you see myo or sarco, that means muscle. So this is your heart muscle, right? So this is where we have that specialized muscle that we were just talking about, right? We did muscle. And then we have a layer of epithelium, and it's called the endocardium, right? That endocardium is continuous with the endothelium, the inner layer of the blood vessels. Okay, so this is continuous. This will then form our blood vessels, all right? Now, I love this dissection because you can actually see the layers nicely. You can even see the space, right, between the different layers of the serous pericardium, right? So yeah, this is just because it's cool. All right, now, remember your heart muscle. All right, I'm not going to go over it again in detail, but remember that these are branched cells, they're striated, and very importantly, in between each cell, we have those intercalated discs, right? And this is a beautiful picture of them, you can see them here. What do we have in those intercalated discs? Well, we have adherence junctions, so junctions that keep the muscle cells together, but we also have gap junctions. Don't forget about that. Those gap junctions and that intercalated discs allow for the movement of the electrical impulse directly from cell to cell without needing nervous support, all right? So this brings us to the flow of electricity through this muscular heart, okay? So if you think back, all right, if you think back to what we had discussed about the cells that are autorhythmic, right? So I said that we have these autorhythmic cells within the heart, and those were called the pacemaker cells. And I had showed you videos of how these guys can contract all on their own. They don't need a nervous connection to do so. Now, these are clustered into two areas of the heart called nodes. So one of the nodes is called the SA node or the sinoatrial node, and the other one is the AV node or the atrioventricular node. And the placement of these and the connections between them are very important for the proper functioning of the heart, okay? And I want you to understand that you can't just have this heart contracting, you know, willy-nilly. There has to be a very controlled style of contraction, and I'm going to go over this very carefully, all right? So remember now that these pacemaker cells, they're going to control the contraction of the rest of the muscles, the muscle cells. So they're connected with these gap junctions. These send the wave of depolarization into the neighboring muscle cells, which will contract in rhythm with them, okay? So it's going to fan out from the nodes, and so we'll start here at the SA node, okay? So we have our electrical activity here. These cells are going to contract, and through the gap junctions, they're going to send electrical impulse, all right, and a wave of contraction across the atria. So a flow of electricity, we start at the SA node, then it flows across the atria. Then we've got a line towards the AV node. The AV node then is going to send its rhythm out afterwards, okay? So the AV node, its rhythmic behavior is a little slower than the SA node, all right? So the AV node now is going to be energized. And very importantly now, this AV node is going to control the contraction of the ventricles. Now, we can't have the ventricles contracting from up here, right? So what would happen if the ventricles contracted up here at the border between the atria and the ventricles? Well, the blood would get squished down into the apex, which is not what we want. We want the blood to get pushed out of the ventricles towards the body, right? So it needs to go from the apex towards the base. So how do we get that electrical impulse down to the apex? Well, what we have then is a flow of electricity through the AV node. It goes down this little thing called the AV bundle, all right? And then it's going to branch because we've got two ventricles, right? So it's going to branch, okay? And these are called the bundle branches. And then eventually it's going to come to the apex. Now, at the apex now, we have to fan that electrical impulse over these huge ventricles. So we do that using these fibers called Purkinje fibers. And that electrical activity is going to go there. And that's then going to send a wave of electricity over the ventricles, which will now contract from the apex to the base, pushing the blood out, okay? So remember that sequence, and you know how much I love sequences, right? So, but it should make sense to you, right? We're starting contraction. We have to get the atria to contract first. So the SA node is going to sort of instigate that, right? We've got the gap junctions that allow the electrical impulse to fan out over the atria. Then we got to get that impulse down to the bottom of the ventricles, down here to the apex. So this is, we get the stimulation of the AV node. The AV node is going to send that down through the AV bundle, the bundle branches, then the Purkinje fibers to spread it out through the ventricles, which are going to contract from apex towards the base, pushing that blood out, okay? Okay, so hopefully that makes sense to you. So now the cardiac conduction system, let's go over that. SA node depolarizes, all right? And I like the color coding here so you can sort of see that wave of electricity spreading over the atria. Now this is now going to go towards the AV node as well, okay? And this, we have these little pathways that help guide that there. And slowly now we're going to get depolarization moving down through the AV bundle, right? Down into the bundle branches, down into the Purkinje fibers. Then as it goes up through the Purkinje fibers, we can see it's then going to go over the ventricle, and we're going to have contraction, right? So you can watch this happening on this video. So let's go over that again, right? So the SA node depolarizes. The activity goes through rapidly toward the AV node, right? We've got internodal pathways that allow this to happen, okay? And depolarization is going to spread slowly across the atria, but the AV node has already been stimulated, all right? This depolarization is going to go through the ventricular conducting system towards the apex. So what does that include? Well, it concludes our bundle branches, our AV bundle, our bundle branches, Purkinje fibers, okay? And then it's going to move up from the apex toward the base, and you're going to have the depolarization wave going upward from the apex, and that's going to push the blood out, okay? So hopefully that makes sense to you. All right, so how do we manage that, monitor that? Well, we have this thing called the ECG or the EKG, okay? And there's a bunch of intervals and waves here. For 105, we want you to know what's happening with these waves, all right? So the first wave on the ECG, that is the P wave, and that is that impulse of electricity going across the atria, and that's going to lead to the systole, or the contraction, all right? So systole means contraction of the heart, so contraction of the atria. Then we have the movement of that wave down through the septum and around the ventricles, and this is then going to just instigate the contraction of the ventricles. all right? So the QRS complex is what that's called, all right? Then the last wave, the T wave, is the cessation of electrical activity. So we have sort of a relaxation period now, and that's recovery in the relaxation of the ventricles. So that would be ventricular diastole. So remember, relaxation is called diastole, and contraction is systole. All right. Now, the one thing that we don't see here is atrial systole. And the reason we don't see it is because that wave would be happening at the same time as the QRS complex. So it's kind of hidden by that really large wave there. Okay. So we can actually monitor this electrical activity and see what it looks like normally and what it looks like if it's fast or slow compared to other patients or irregular, which is why it's really important to get an ECG done. And then that they can compare your ECG at different times of your life to see how things are happening. Ventricular fibrillation is when things are not working well at all. We're not having proper conduction, all right? So what they try to do here is to reset the pacemaker cells. So they apply the electrical stimulation with the defibrillator and try to reset that and get the heart working on its own again. Okay, so clearly if this is happening, if the conduction of the electricity isn't working well, obviously it's going to lead to death, so heart attack. Now, the cardiac cycle, all right, so this now is talking about the flow of blood in response to this electricity, all right? And in 105, we just sort of divided into 3 steps. If you go into AMP, you're going to get all 5. But here, for the cardiac conduction cycle, we'll just go over the flow in these 3 major steps. So remember, systole or the systolic is contraction, diastole is relaxation, all right? So the first thing, if we always start from the atria. So the atria are going to contract. We've got atrial systole, and that's going to force blood through those AV valves, so the bitricuspid valves, into the ventricles, all right? And at this time, the AV valves are open. Then we're going to have the ventricles contracting. So we've got the electrical activity going through the AV bundle branches, Purkinje fibers. Then we'll have the ventricles contract. So what's going to happen now is that the AV valves are going to snap shut. All right, so that's your first heart sound. So those valves snap shut, and blood is going to be forced out through the semi-lunar valves, so the aortic and pulmonary valves out towards the body. Then what's going to happen is the third step. So ventricular diastole. So the atria are going to, the ventricles are going to relax. When they do that, that back pressure is going to snap the semi-lunar valves closed, and that's going to be your second heart sound, so your dup sound, all right? And then we reset ourselves, we come back to the beginning, all right? So if you go into AMP, you're going to have another couple of steps in here, all right? But for 105, those are the main parts, and you should at least understand what's happening there, okay? Now remember that when the blood flows out, we have the expansion and contraction of these elastic arteries, and that we can use to monitor blood pressure, all right? So the way that works, and we did it in labs, just remind you, so the blood pressure cuff is going to cut off the blood flow, and then as the cuff starts to relax, okay, the first sound that you hear is the spurting, and that will coincide to ventricular systole, right? So when the blood is being pushed from the ventricles, all right? So that's your higher pressure, your systolic. And then eventually you're not going to hear the squirting or the spurting sound anymore, and that will be your lower blood pressure, your diastolic pressure, and that's when the sound is a rush, all right? And you can hear this on any of these elastic arteries. So we've got the brachial artery, the radial artery, the common carotid artery, and then in the leg, the femoral artery. You measure all of these elastic arteries and detect this pressure, all right? And we use this device, a sphygmomanometer, to measure that, okay? And that's measuring this pressure. So the high pressure is the systole, so that's ventricular contraction. And then the lower pressure is diastole, or the diastolic pressure, and that's ventricular relaxation, all right? Okay. So things that can go wrong, right? So we have hypertension. So that is high blood pressure. So that's when those pressures are higher than normal. Hypotension, right? That's low blood pressure. Um, Different people, depending on their age and their size and their sex. So hypotension is really going to depend on your symptoms. So if you're fainting and that sort of thing. Hypertension, though, we have pretty solid measurements on that, okay? And it's divided up. You don't need to know the numbers, but I just put the chart here so that you can look at it. You know, your systolic and diastolic pressures for normal, pre-hypertensive stage 1 and stage 2, right? What are the risk factors? Well, there's a lot. So some things you can't do anything about, right? Heredity, age, race, sex, can't do anything about that. Other things you can, obesity, high dietary salt, smoking, how sedentary you are, how stressed out you are, diabetes, alcohol, contraceptives. These are things that you have some control over. What else does hypertension affect? Well, vision, stroke, dementia, headaches, blood vessel damage, which we'll talk about a little bit, renal failure, because you've got lots of capillaries there. If they're under high pressure, that would be devastating. Heart attack, right? So when it comes to this, and we're talking about these pressures, how does the body regulate these pressures? And the three things to remember, right? So clearly you can see how high blood pressure affects so many different parts of the body. So you can see that homeostatic regulation of the CV system is really important. And the more constant your arterial pressure is, the more homeostatic you're at, okay? So how do we control that? Well, we regulate our cardiac output, so how much blood is being put out by the heart, and also the diameter of our arterioles, right? And then there's also a regulation of local blood flow, all right? So how do we do that? Okay, so it's a negative feedback system, all right? So we have sensors for pressure in the aorta, and that's going to be monitoring pressure out to the body. And then we also have pressure sensors in the carotid arteries, and that's going to be measuring pressure going to the brain, all right? Now, these sensors are called baroreceptors because they're sensing pressure. So remember, the barometer is monitoring air pressure. So the same thing here, the baroreceptors here. And so they're monitoring this pressure. And they will send information to the brain depending on what's happening. So we have these sensory neurons. They are going to respond to an increase or decrease in pressure. They send more signal with increased pressure, sends that to the brain, and then the brain is going to respond. It's going to send information to the blood vessels, and it's also going to send information to the heart, okay? And then when the blood pressure increases or decreases back to what it should be, or what's normal, and I'll come back to that thought in a sec, then you know, we've got our negative feedback. So if we look at this a little bit more carefully, so say the pressure is too high, all right? So what's going to happen is that we're going to stimulate the region in the brain. It's called the cardioinhibitory center, right? It's going to try to inhibit. It's going to send information to the heart to decrease the heart rate. And this is kind of weird, all right? So to decrease the heart rate, what ends up happening is that the neurons are actually going to send more stimulation, right? I'm going to talk about this later when we talk about the nervous system. But what's happening here is that the SA node actually has a rhythm that's higher than what the body likes. So it's easier for the brain to just sort of dampen that down, and that's what it's doing, okay? The other thing that's going to happen is that the vessels are going to dilate. So remember, as they dilate, that's going to lower the pressure. So then the pressure is going to come back down. Then when there's a decrease in pressure, all right, the cardio acceleratory center, okay, is going to come in and have its role. So what it's going to happen then is that there's going to be a sympathetic stimulation of the heart. And what's going to happen here is actually the brain is going to sort of pull back and then let that SA node do its thing, right? So the heart is going to beat faster. It's going to increase the cardiac output. We're going to constrict our vessels, increase the pressure, okay? And then we allow for the pressure to come back up again, all right? So during high pressure, stimulation of the pacemaker cells causes them to slow down. And then during low pressure, reduced stimulation of the pacemaker cells causes them to speed up. And that's because the pacemaker has a very higher resting rhythm than what our actual output is, all right? So it sounds weird, but that's the way it works. And then, of course, when regulating at the tissue level, the precapillary sphincters can be regulated to control the pressure going through the tissue, okay? And these are paracrines that are released by the tissue itself, all right? Now, things that can go wrong, angina, all right? So this is where we're having disruption of the coronary circulation, and that causes pain, right? And you can sort of see it in an angiogram. You can see it there. It's sort of dark and the flow isn't going through. Then we have heart attack, okay? So that's where the coronary circulation is actually blocked and we have death of the cells, all right? So that's the heart attack. Then we have heart failure. Now this is different, okay? So what's happening here is that the heart muscle becomes weakened, all right? And there's different reasons that this can happen. Very often it's a valve issue, for example. And what the heart does is it tries to compensate for that, all right? So the heart can enlarge in order to try and be more efficient with its pumping. So it tries to get stronger. And the thing is that the pressures then become a new normal and the body just kind of gets used to it. So remember, we're talking about this process here. This is how we can, increase the pressure and then the body just kind of gets used to it. And it can also be the other way around. So the body tries to get to its new normal. Now, what's happening here now is that with heart failure, this eventually does lead to death, okay? And it can happen over several years, all right? I've witnessed it. It happens to be the way my father passed away. But slowly, it leads to this idea called congestive heart failure. So the heart just in general becomes less efficient. And even though it's beating, all right, fluid is going to start to back up in the tissues. So it's going to back up around the lungs. The kidneys aren't going to work properly. It will pool in the ankles and other places. And as it pulls up around the lungs, it gets more difficult to breathe. And then, of course, there's less oxygen. And then, of course, then there's that message to have the heart to move, beat a little faster. So, because there's also sensitivity to how much oxygen is in the blood as well. So it can, and like I said, eventually it does lead to death. The whole system starts to break down. And this is where I start talking about my holy trinity of organs, which is the heart, lungs, and kidney. When one starts not working right, the other two eventually will be affected. And they are connected, obviously, through the circulation, right? So that's that idea. So the other thing is embolism. So this would be a sudden blockage of a vessel. And these emboli are often given by their name. So cerebral embolism, which could cause a stroke, so blockage of vessels there. Cardiac embolism could be precipitating in heart attack. And stroke, of course, then is damage due to the loss of circulation to the brain, all right? And this can also be because of a ruptured artery as well, okay? So think of, a cerebral aneurysm, for example. So that can happen as well, all right? So stroke could be to embolism or back to our idea of aneurysms, right? I said most of the aneurysms are happening in the aorta, but they can happen other places as well, all right? So a brain aneurysm. Now, so things to keep in mind for your cardiovascular health. Well, number one, don't smoke, all right? Smokers have twice the risk of heart disease. Watch your cholesterol, moderate exercise regularly, monitor your pressure, treat any hypertension. Try to maintain a healthy weight for you. Okay, that's going to be different from different people. Keep your diabetes under control if you've got that, and try to avoid chronic stress. I know in our times it's difficult to do that, but you've got to try your best. Okay. Okay, so I think that's about it.